welcome everybody again to the Noyo Center Science Talk. Tonight we have a guest that I'm super excited to hear a presentation from. Rietta Hohman and I have been working together since the kelp story began to unfold. Way back when we were first working with the divers to remove urchin. Rietta was part of the brain trust that was putting together the initial sort of steps and figuring out how we tackle this incredibly fast change in our ecosystems that we were seeing. And one of the things that she did that was so significant way back then was bring together all the science community that had a role in kelp forest ecosystems to create the first kelp recovery plan. A year of meetings to pull together a state of knowledge, which was limited. I think everybody would say that. But now Rietta has continued to build an impressive program around kelp restoration for the sanctuary. So I'm going to start off with introducing Rietta, who is the Kelp Restoration Project Manager for the Greater Farallones Association and the sanctuaries, which have now combined to become the Greater Farallones and Cordell Bank National Marine Sanctuaries. As I said, Rietta's been working on the kelp forest issues from the beginning and has been one of the leaders in this this effort from the Mendocino and Sonoma coast for sure. She has put together an incredible team and many collaborative projects working on various issues, which you'll hear much more about tonight. I was just on a meeting with her not too long ago, which hopefully we'll talk about a little bit, because as you know, Noyo Center is really putting their efforts into some of the conservation aquaculture around purple urchin, restorative seafood, but also looking at trying to do an abalone broodstock program and grow some seaweed as well for some of the work that's being done. So our paths are intersecting because the work we're doing on abalone is with the Kashaya tribe, and part of the first phase of that abalone broodstock program is to remove urchin so we can get some habitat to put abalone back into. So that's where we came back to Rietta's doorstep and said, how can we work together? And you'll hopefully hear more about that tonight. So with that, I want to introduce Rietta Hohman. Thanks, Rietta, for joining us. We really appreciate your time tonight. Take it away.
Okay, perfect. Thank you so much, Sheila, for a lovely introduction. Really excited to speak with everyone tonight. I'm going to be talking about our project, which is the restoration of kelp forest habitat in Greater Farallones National Marine Sanctuary. And as Sheila mentioned, this is a partnership between the Greater Farallones Association, which is a nonprofit, and then Greater Farallones and Cordell Bank National Marine Sanctuaries, which are part of a federal entity under NOAA. And so it's a joint project. It's a really unique partnership. We obviously have a lot of partners that work with us on our kelp restoration project, but I'm going to be speaking from the perspective of this joint project. So just to give a little bit of an orientation on our sanctuary. So our headquarters is actually down here in San Francisco. Not sure if you can see my mouse, but we're just at the foot of the Golden Gate Bridge is where our office is. And then the Greater Farallones National Marine Sanctuary encompasses about 3,200 square miles of protected ocean space on the Northern California coastline. And then we are a joint sanctuary with Cordell Bank as well. So it's all managed by one group of people working together. And then our kelp restoration sites are identified on this map. They're circled in red here. So we have 3 sites that are in the area of Sonoma from Fort Ross Cove up to Ocean Cove. And then as of this year, we have this new collaboration that's launching with, Sheila mentioned, the Kashaya Band of Pomo Indians and the Noyo Center for Marine Science, as well as UC Davis, that we're really excited about. So I'm going to be talking just mostly about these sites in Sonoma and then just touching base on this other project. So this is zooming into our sites in Sonoma, and the work is happening that I'm going to be talking about today is mostly happening at Fort Ross Cove and Timber Cove, if folks are familiar with that stretch of the coastline. And then this year, we're going to be starting work at Ocean Cove. Now, Stillwater Cove, we're definitely interested in doing work there, but it's not currently funded. So I'm not going to be talking much about Stillwater Cove, but it is definitely one of our sites of interest. Now, I know a lot of folks are quite familiar with the kelp decline on the North Coast, but just to kind of set the stage, the background for the work that I'm going to be talking about resulted from the catastrophic decline of kelp forest habitat that occurred in 2013 to 2014, and that has persisted since then. So we've seen no significant recovery of kelp in the past decade. We did see the collapse and closure of the recreational red abalone and commercial red sea urchin fisheries. In 2018, the Greater Farallones Sanctuary Advisory Council formed the Kelp Recovery Working Group, which was a very diverse group of stakeholders, researchers, nonprofits, tribes, different folks who came together and really looked at what was happening on the North Coast, what data sets we had, and how could we develop recommendations for management, both the state and the sanctuary, to come together to implement for kelp restoration. And so coming out of that process, that's where we developed the kelp restoration plan for Sonoma and Mendocino. And then more recently, these past couple of years, we've seen some funding awarded from state and federal sources for the Kelp Restoration Project. And so our project is focused on restoring these areas where we've seen this significant drastic increase in purple urchin densities, and they've now become urchin dominated, to return them to healthy kelp forest habitat. So that's our goal. And we really want to see this throughout the sanctuary, but we're focusing our efforts on a few key areas for a particular reason that I'll talk about in a moment. So our project, I know there's a lot of acronyms up there at the top, but that's just Greater Farallones Association and then the two sanctuaries, Kelp Restoration Project. So our goal, as I mentioned, is to restore kelp forest habitat in the sanctuary. Our objective is to focus a multi-pronged restoration approach at those three sites, which is now expanded. So I'll talk more about that in a moment. But we're really focused on reducing urchin population densities to allow breathing space for kelp to grow. We culture and outplant bull kelp, and outplanting is quite relatively new in the field of restoration as far as kelp is concerned. So we're learning quite a lot in that process, but we're very excited about doing that work. And then doing a pretty robust monitoring and assessment process so that we can better understand how to conduct restoration and how to have an adaptive management approach. So, oops, went backwards. So our restoration site selection, this was a big part of what the Sonoma Mendocino Bull Kelp Restoration Plan addressed. But what we looked at was historical persistence of kelp canopy cover. So that's what you're looking at. This bottom right-hand image is showing Fort Ross Cove. And what you see here is persistence of kelp canopy. So the areas where it's lighter, that is fewer years, 1 to 2 years of kelp cover. And then as you get darker and darker, you see more and more years of kelp cover. So basically dark colors mean that kelp occurs more often, so there's higher persistence. And so this data comes from the California Department of Fish and Wildlife. And then we've also looked at Landsat data, which is what you see here on the left side of the screen. Landsat has this really amazing 38 plus year time series of kelp canopy data that we can look at to really see where there's persistence of kelp canopy cover. Now, the reason that we want to look at these is because we want to focus our restoration efforts where kelp is most likely to grow. So we want to target these restoration areas that are really dark, but not these areas where we've never seen any kelp cover. So this is very, very important. And then I'll talk about this in a few minutes, but we also go out to all of our restoration sites and we map them with drones, which gives us very high resolution kelp canopy cover, which is really exciting. So these data are incredibly important for restoration site selection. We also looked at a whole bunch of different things, including economic and cultural value, accessibility, and historical data sets, like where there's been PSCO surveys, for example. So that all factored into our restoration site selection in the sanctuary. So this is our team. These are our rock stars. These are the folks that go out and do all of our surveys. They spend hours underwater. They're really an incredible team. If you take a look, this is actually on the right. This is our team out on the research vessel Fulmar, which is a federal vessel that is provided by Sanctuaries for us to do our work. And then on the left, this is the small boat from the Fulmar. So this is a Sanctuaries vessel. And so Angela, Gina, Julieta, and Tyler are our team. So they're not just divers, but they also manage different aspects of our project, including the urchin removals, outplanting, and monitoring and assessment. So our urchin removals, we're just going to dive right into it. So we worked last year, this is 2023, we worked with 5 local commercial urchin divers, local meaning out of Bodega Bay. GFA, which is the nonprofit, has a fish buyer's license. So basically that means they can purchase urchins from commercial urchin divers. We work with a local family-owned business called Dandy Fish Company. And Dandy offloads, weighs, and transports the urchins to a compost facility. So they handle kind of everything once the urchins get off the boat. And then last year, we had a total of 80 diver days that divers were out on the water diving and removing urchins, which totaled about 260 hours underwater. And all total, they removed around 24,000 pounds of urchins from our 2 sites, Fort Ross Cove and Urchin Cove. Urchin Cove? Fort Ross Cove and Timber Cove. So that was a pretty incredible effort, especially because we had a lot of delays last year because of permitting challenges. But this was a pretty significant haul from the commercial urchin divers, and it was a huge learning process for us. And it worked out really well. And so you can see our team actually counting and measuring urchins down here on the bottom left. And then Dandy weighing the urchins on the right. And then one of our commercial urchin divers, Eric, here with our team member, Gina, with his urchins. So if we take a look at Fort Ross Cove, this is just showing our general restoration area. So we focused our efforts in that middle area of the cove. Then we had some restoration infrastructure set up to help guide some of those urchin removals. And then we had these alternative, more protected areas so that when the weather picks up and it's really tough to dive out there are these areas around the restoration site that they can remove urchins to keep that work going. And then at Timber Cove, I know this image is a little distorted, but this is just showing a kelp canopy cover at Timber Cove, and then where some of the urchin removals happened around that those kelp canopy beds. And so the approach in Timber Cove was to protect emergent kelp or kelp that was growing. Now, we did see a pretty significant reduction in urchin densities, of course, in areas that were uncleared versus cleared. So here we can see the urchin densities in Timber Cove were reduced from 20 urchins per meter squared down to around 4 urchins per meter squared approximately. Our target is 2 or less urchins per meter squared. But we had like I said, a pretty limited time window last year. So, but less than 2 is our target density. And then we don't target reds, but we do count red urchins. As of right now, reds are not the actual focus of our efforts because the purple urchins are the ones that are in higher densities. And then we saw, for folks that are familiar with gonad index or not familiar, what that means is just the ratio of gonad to the body size. So it's kind of a gauge of how healthy an urchin is. Now at Timber Cove, there was already some existing kelp. And so we know that the urchins had more food there. So we were much more likely to see a higher gonad index at Timber Cove than at Fort Ross Cove, and that is what we saw. So that aligned with what we were thinking. And then what we would expect to see over time during our restoration work is that this gonad index would increase, right? Because if we're doing restoration at these sites, we would hopefully see more kelp growing, which means more food for urchins. So hopefully they become healthier over time. So we will, this was to lay a baseline, but we will be tracking this throughout our project. Okay, so we've worked really closely with Sonoma State University and with Moss Landing Marine Labs to establish a culture facility at Bodega Marine Lab. And so what we have there, we actually have an office there and spaces that we rent. But we've established culturing tanks where you can see our two scientists here. On the left is Rietta Hohman, which is one of our kelp restoration specialists. And on the right is Rachel Carm, who's a research assistant with Sonoma State University. And so they've worked together with our respective teams to establish these cultures. What you see here is our tumble culture. So what they're holding up are bull kelp crowns, which just means the very top of the nematocyst or the float that's cut in half, and it has all of the blades still growing off of it. So we put a whole bunch of those crowns in a tank and put them through a tumble that's oxygenated so that over time they'll eventually release spores. And so those spores are then collected and put into spore bags. And the spore bags are then outplanted at our sites. And so far we've only outplanted at Fort Ross Cove, as well as Drake's Bay, which I'll talk about a little bit more in a moment. And then what you see on the right, this is the other method of culturing that we use. This is a PVC pipe that has twine, either synthetic or a natural material twine that's wrapped around it. And then it's cultured in water that has spores from bull kelp, and those spores will settle, grow, and become sporophytes. So you see these little baby kelps that are then growing on the twine. And then on site, we'll take these out to our restoration site, unspool them, and then they'll be affixed to the outplanting site. And I'll show you what the outplanting site looks like in a moment. But then the idea is that they would grow from the twine and then become reproductive adults. So that's what's happening at Bodega Marine Lab. And this is really the only infrastructure in our area that is possible, or where it's possible to grow this much kelp. And so it's really been this fantastic adventure to establish what we need at the lab and to consider how we can help support our project moving forward with these culture facilities. So this has been really exciting. This is what the outplanting site actually looks like. And I know it's a little bit of a funky diagram, but what you see on the top left, this is Fort Ross Cove. This grid is part of our restoration infrastructure. So this is like bolts and lines, lead lines, that we set up underwater. And we only set up this one part of the grid this past year because it's all that we needed in the time being. The rest will be set up in the future. And then each one of these blocks is separated into quads. And now these are not actual lines and bolts in the water. They're just kind of how we've separated them on diagrams. But then at Fort Ross, this A3, so this bottom kind of left-hand corner here, is where we've outplanted bull kelp. So these little T modules are set up with spore bags, seeded lines, a control line, so just a blank line that doesn't have any kelp growing on it, invert recruitment modules, and then what we call pavers. And I didn't talk about pavers as much because we're not going to be using those moving forward. The pavers are basically these little 3-inch by 3-inch concrete blocks. And the blocks are very heavy. They take up a lot of space in the lab. So we will not be using the concrete blocks moving forward, the pavers, just because it's a lot of effort to outplant them. So we will be using the spore bags as well as the twine in the future. So this is just kind of in general what the outplanting setup looks like. Now, we have outplanted in Drake's Bay. This is kind of a project that's wrapping up right now. So we're in the process still of wrapping up all of the data. We're probably going to do one more survey. But we've been working in Drake's Bay for the last couple of years. And Drake's Bay is around Point Arena, just north of Bolinas is our restoration site. And so the outplanting methods that we've used out here is the twine that I showed a moment ago that's cultured with bull kelp sporophytes. And the twine is actually wrapped around the clay bricks, and the clay is then placed into the water. So you can see this first photo, the twine being wrapped around the brick, and then the brick being placed in the water. And so that brick would then be weighted, go down to the bottom and provide substrate for the kelp to be able to grow from the bricks. So that was a really great project that we've been working on, like I said, for a couple of years. And then we did team up with Fish Bio and Monterey Bay Seaweeds. This is Dan Gossard and Elizabeth. And what they did is put together this concentrated zoospore solution where they could collect bull kelp the day before and then have a solution 24 hours later. And then that bull kelp zoospore solution was put into this backpack and then basically pumped down to the reef. So it was like a reverse vacuum. So it was pumped down to be dusted over the reef. And so we use these couple of outplanting methods in the area. And like I said, we're still processing data, but we did see over the 2 years an increase in kelp canopy cover of about 5% across all of our sites. So that was really interesting. So TBD on this project, but we're wrapping it up in the very near future.
Quick side note that we did get a science talk from Elizabeth on the reef duster, if anybody wants to go back and watch it.
Oh, excellent. Yes, they will definitely have a lot more info about it than I will.
Yeah,
but we're very excited for
yeah, it's very cool.
potential future applications. Okay, so our team that I showed a moment ago are super busy during the field season doing a lot of monitoring and assessment. So we have a regime of subtitle ecosystem and restoration assessment surveys. So our team are all NOAA divers or trained by NOAA to do this work. And so we use PISCO protocols mostly, and then adapted protocols from the state of California to measure urchin densities as well as bull kelp stipe density. So we'll go out and track the urchin removal efforts throughout the season. And then the ecosystem surveys are kind of these set locations at a restoration site. We do those annually. So it gives us a snapshot of the whole ecosystem health. And then we sample a ton of urchins. So when the urchins are landed, we will be at the dock and basically pull a few of them from each site into a cooler and then take those urchins to the marine lab and capture data on their morphometrics, or just basically like their test diameter, the height, and how big they are. And then look at the gonad health. So we'll actually dissect them and look inside of them. So we'll sample those from each site. We do kelp canopy surveys as well. As I mentioned a few minutes ago, we have drones. So we'll go out to the coast and map with drones, and I'll show a little bit more about that in a moment. We do beach rack surveys, which is just going out to the restoration site beaches and adjacent beaches, and then capturing morphometrics of bull kelp that is washed up on shore. And then we have a new project that's launching this year to do intertidal surveys at Fort Ross Cove, and hopefully urchin removals as well, kind of coming into some permitting questions. So this is our team getting ready to go underwater, and then some folks that are actually underwater. So the subtitle surveys, if you remember our graphic from a moment ago, this infrastructure is actually super, super helpful for helping guide our monitoring efforts. So this is the same block that I showed a moment ago. but then our team will go out and they have fixed locations that are along that line, that lead line that's fixed underwater. And they'll go out and they'll do a 30-meter by 2-meter transect. That involves laying out a transect tape with 2 divers, and then the divers on each side will actually count and measure urchins and bulk kelp density on both sides. So that really helps us capture robust data over time. And we'll do these across each one of these quads. So this is a lot of surveys, but it's really important for us to help capture that data and information over time, because we can help guide the urchin removals. We can say, hey, you need to come back to this area, or the urchin densities are still a little bit too high. And then we can better understand things like encroachment. So like urchins moving into a space after it's been cleared. And then just better understand ecosystem recovery as well. And then this is our research vessel. This is the Fulmar, which we use in the fall each year. Okay, so these are our beach track surveys, or some of our team doing the beach track surveys. This also involves laying out a transect tape. And then we have quadrats, so they'll go along and actually look at what beach track has come up or washed ashore. And then if there is bulk kelp, we'll capture data on morphometrics, like I said. So this is something very new that we've been doing over these past few months, but we hope that this could be a really cool opportunity for citizen science engagement in the future, or volunteers that want to work with our project, because this is a really accessible way to get out and do a lot of data collection and participate in the project where you don't have to have a scuba certification. So this is something that will hopefully expand in the future.
Well, Rietta, let's be sure to talk about that, because we have a beach survey team that is actually collecting some of that data and has been for years. And so it'll be great to make sure that we're aligned and collecting the same thing.
Yeah, definitely. And we go out to these sites about once a month, I think. So we are trying to lay a baseline for the entire year. That way we can capture what the changes are over time at our restoration sites. So this is hopefully laying a baseline to help us better track and monitor restoration. Okay, so this is the new project that I mentioned. So we will be doing intertidal surveys and eventually urchin removals pending permitting at Fort Ross Cove. So this is Fort Ross Cove here. And you can see some folks on our team just being silly at the site. But we are currently monitoring this site. So we go out and we have transect tapes and we count urchin densities. And then the idea is in the future, we'll be doing some removals here, and that will help the intertidal community here recover, because the urchin density in the intertidal is also very, very high. It's not just the subtidal, the intertidal is very high. And we want to understand the encroachment rate, encroachment meaning from the subtidal up into the intertidal, and if that could potentially aid with our urchin removal efforts at Fort Ross Cove. So this is a new project launching. We'll see how it goes. And then you can see here just how dense those urchin populations are. This is Fort Ross Cove in that same area. And then we can see there's quite a lot of life here. There's abalone, sea stars. So this area, we're really interested in seeing how this community might recover over time. And then we work with Fort Ross Conservancy. They're going to be helping develop some materials for our project and a curriculum to help teach youth and visitors, the public, about kelp restoration at Fort Ross. And so we work with the Conservancy and then also with our sister program with Sanctuaries, which is Limpets, for long-term monitoring and experiential training for students. So our colleague, Jacqueline, who's right here, takes students out to these intertidal areas and captures data in the intertidal on invertebrates and algae. And so we work with them to establish these sites. There's one at Fort Ross Cove, and then there's one at Gersel Cove as the control site. So we're hopefully going to be expanding those outreach and engagement opportunities in the future as well. Okay, and then as I mentioned earlier, so we have a pretty robust effort to get out onto the coast and map kelp canopy, which helps us track changes at our restoration sites over time. And then on a larger scale, helps us monitor kelp forest recovery in the sanctuary. So we, in the kelp restoration plan, identified about 27 sites in the sanctuary that were of high priority for kelp monitoring efforts. And that's a lot of manpower, person power, to get out and actually map those sites. And so we've had to narrow it down to about 10 to 12 sites where we actually get out and map them that are adjacent to our restoration sites. But this is really critical information for our project. When we can't get underwater, we really rely on this kelp canopy to inform us about how our restoration effort is going. And so kelp canopy data is super duper important, and it also helps us understand dynamics and trends. We can look at that Landsat data, that state collected data, and understand how things might be changing over time. This also helps us focus our urchin removal efforts, because if we see there's a kelp bed over here at Timber Cove, we can direct the urchin divers to this area to help protect that urchin or help protect that kelp bed. So that's one of the projects that we've been focusing on, is protecting existing kelp or emerging kelp and allowing it space to grow so that it can continue to produce spores for the surrounding environment. So kelp canopy mapping is really important. And what you see here, this is an orthomosaic on the top here of Timber Cove. An orthomosaic is basically a whole bunch of different images that are stitched together. So when the drone, you can see the drone here in the bottom picture, goes up about 400 feet in the air and does a survey, it basically takes thousands and thousands of pictures. So all of those pictures are then uploaded into a software like Drone Deploy, and it automatically stitches them all together and makes this orthomosaic, which is like a georeferenced image. So this is basically the product that comes out of that kelp canopy mapping. That is incredibly important for our project. So that's the orthomosaic. This is our team. We're working with the Kashaya Band of Pomo Indians at the Kashaya Coastal Reserve. And then on the right here, this is one of our volunteers working out at Timber Gulch. So I do want to just touch base. These last couple of slides here, I'll kind of focus on some of our challenges that we've encountered. And those are kind of extensive. This is the short list, but there are a lot. Accessibility is huge. So these sites, we mostly chose them because they're accessible by shore and by vessel. There are a lot of areas of the Sonoma and Mendocino coastline that are not very accessible at all. So accessibility is really big. Equipment reliability and issues. We've encountered a lot of issues with small boats. Our ROV, which is a remotely operated vehicle. We have this little drone that has a camera on it that we'll send down to do surveys. Our aerial drone during the Drake Spey cruise literally crashed into the boat and broke. And so all of those things can cause delays. They can cause loss of data. They can kind of result in a lot of different significant challenges, and we've encountered all of them at this point. So you kind of just have to know that that's going to happen and be able to maintain flexibility in the field. I think that's been one of our kind of biggest lessons learned, especially from Drake's Bay. We always encounter really dynamic weather and ocean conditions. I know probably most folks are quite familiar with that on the North Coast. it can be tough to get out there. Right now, it's super windy, so our urchin divers cannot get out to their sites. And so ocean conditions and dynamic weather can really be a pretty significant hindrance to getting to the restoration sites. Now, I mentioned outplanting really is quite new. These are novel kelp restoration techniques that we're using. Many of the techniques have not been used in other areas. We don't know how effective they are. We don't know the success. We're really just starting out a lot of this work. I think we've seen some really great progress in some ways, and then other ways we just don't know. And we've also learned things that definitely don't work. So as of right now, outplanting is tough to use as an actual restoration technique. It's still very much in a research phase. So that is pretty important to consider, especially when scaling restoration efforts. Identifying new liabilities and obtaining insurance. I forgot this one was in here. But this is actually a huge one for the urchin removals. When we were initially working with urchin divers, it was really tough for us to figure out how to work with them because we were going to contract them, but there's pretty huge liabilities in contracting commercial divers. So we eventually developed a model that just worked with the divers in terms of us purchasing urchins as fishmongers or having a fish fire license, and then they're working under their own commercial urchin license. And then developing that framework from urchin removals for restoration sites by commercial divers. We've had a lot of trial and error in this respect, but I think we've developed a pretty solid model and have been pretty successful at implementing this. And we're excited to scale up and apply these lessons learned to other areas as well. I know this is not super exciting, but I have to touch base on it because it basically consumes much of my life right now. But environmental compliance is tough, especially when you're looking at marine restoration. Environmental compliance is pretty crazy. There are multiple layers of federal and state permitting, permissions, and approvals that are necessary. So we're working in a national marine sanctuary, which is huge. And so the sanctuary regulates anything that touches the benthic environment or that is considered discharge, which outplanting is, because it's introduction of material, even though it's natural material. It is an introduction of material into the water, so it is regulated. So we work in the sanctuary. We also have to consult with National Marine Fisheries Service, National Park Service. We have a NEPA analysis that we have to do as part of our restoration work. We have multiple consultations with the FH, the Endangered Species Act. We have tribal consultations, National Historic Preservation Act. All of those are extensive and they take a lot of time. And so that's why our field season last year was shorter than expected, because these consultations can take months and months and months. And there's no way to like speed up this process. So this is mostly for practitioners that are looking at implementing your own restoration project. Just allow a lot of time for permitting and permissions. And then we also work with various state entities. The California Department of Fish and Wildlife has permits, state parks. We have a state parks permit, the California State Lands Commission, the Coastal Commission, and then CEQA. We haven't done CEQA to date because it hasn't been required for our project, but it's good to know that it exists. And then there are also local permissions that are needed, like county parks and coastal communities, especially when we go out and map with drones. Because for folks that live in like Sea Ranch, if we go out and we map areas, they're just going to see a drone flying over their houses and not really know why it's there unless we get that permission in advance. And so there's just tons and tons of considerations for environmental compliance. And this is not the most exciting stuff, but it is incredibly important. And without all of this, there's no restoration project. So just wanted to highlight some of those things. So next steps, starting to wrap up in 2024 to 2025. So I mentioned kind of at the beginning, we had this really exciting new collaboration with actually the University of California, Santa Cruz. We were funded through a UC or through a California Sea grant to do research on how to remove urchins from around emergent kelp beds. And then we've teamed up with Kashaya and Sheila, as I mentioned earlier. And so we'll be working in the same spaces to do urchin removals as part of that collaboration up in that area. And then we're still very much entrenched in environmental compliance right now, but we are looking to get our work started and have already started urchin removals. And then just in general, as part of that environmental compliance, we're renewing all of our permits to scale restoration efforts and implement several projects this year. Some of which I've already mentioned, but then in general, just to continue restoration at Fort Ross Cove, Timber Cove, and begin at Ocean Cove. We have an oceanographic monitoring project that we're hoping to launch, which involves sending moorings with sensors out at our sites. So TBD on that one. And then continuing outreach and engagement. So those are kind of the big things that we're looking to advance this year, like the high level overview. And I think that brings us to the end here. And I wanted to just say thank you to lots of folks that have helped with various aspects of our project, and wanted to pause for questions. So thank you.
Thank you, Rietta. That was amazing. And it made me remember why we don't lead this work. Your list of challenges. I mean, when I saw your list of challenges, and I'm like, wow, she doesn't even have permitting on there. That's like the biggest one. Little did I know you needed to have your own slide for permitting. It's an impressive amount of work that you guys are doing. I mean, it really is. And the team of organizations you've brought together is super cool. But I have a couple questions initially. We didn't get any questions in the chat while you were talking, so you did a good job covering your topic thoroughly. But my questions are around the oceanographic monitoring, and is that are you coordinating with Senkus, or how are you doing that, or what can you tell me a little bit more about that?
Yeah, so we're working with UC Davis Bodega Marine Lab, John Lerner's lab, to launch or deploy 4 moorings at our sites, our 3 restoration sites, and then Gersel Cove is actually our control site. And then we have a suite of sensors on each one of those moorings to capture pretty fine scale environmental data and oceanographic data. And so we're hoping to implement that over the next 3 years. And then we also have a project that's part of that one, but it has a very specific focus on a hydrodynamics study, working with Scripps Institute of Oceanography and Stanford University. So they're looking at how restored kelp forests influence nearshore waves and currents at our restoration sites. So we're going to implement that this year. All of that depends on us doing archaeological surveys, which are part of our permitting process. And we're having some challenges with those, but that's the landscape and the partners on that project.
Yeah, that's amazing. So that's great. I'm wondering, so I was wondering what the purpose was of some of that, because if you're forecasting some of these changes through the environmental monitoring, the oceanographic monitoring, is that to potentially regulate what you do the following year related to restoration. I keep thinking, like, I want to see more of that oceanographic monitoring here. And then I ask myself, well, but what is the purpose? Like, if a heat wave is coming, or, it's like, what, but you wouldn't want to do a lot of, spore dusting if you knew you were going to get a big fat heat wave, coming, I guess. So
yeah, it's really to help us understand the fine scale site specific oceanographic conditions that are conducive for kelp recovery, because we know that kelp grows at Timber Cove right now, but we've seen no persistent kelp growth at Fort Ross Cove. Is that because of the urchins, or is it because of environmental conditions? We don't really know. And so if we have that environmental data, that will help inform our site selection and our restoration efforts so much. it would just be like this level of detail in terms of collecting environmental data is unprecedented. This hasn't really been done anywhere. So we're really excited to get that project up and going.
Good,
I
good.
think
good.
it'll help
Okay,
us understand a lot.
so you're not talking about large drivers, then you're talking more about local conditions that are creating these
Yeah.
site specific.
We're talking about local
persistent
conditions,
problems. Yeah,
but they will help us understand the influence
Yeah.
of larger environmental drivers.
yeah, very, very interesting. Okay, my other question before I open it up to everybody is about your kelp twine. Like, so we're gonna tumble a little bull kelp ourselves, by the way. We're very excited about that. Gonna be doing that soon.
Mhm.
We've been working with Moss Landing on some of our tumble culture, so we've decided to do a tank of bull kelp just to see, and to hopefully feed the abalone when we get the abalone going. But the twine, you said you wrap it around the PVC pipe and then you take it out and you untangle or unwind it from the PVC. And then how do you secure it on the bottom? And because that twine looks so little from what I had imagined in my head.
Yeah, it looks really small, right?
Yeah.
So it's actually fixed by bolts. So there's bolts that are put into the reef, and then the twine is strung in between them off of the top of the so they're off the reef. They're not resting on the reef.
Right.
So they're held suspended between the two bolts.
Great. So, and then do you go collect the twine or does it just disintegrate in the environment because it's natural?
Yeah, so to date, we've used hemp twine, which degrades really quickly. We've actually learned it's not a good material to use. Note for people who are culturing bull kelp. We were trying to find a biodegradable material, but hemp is just not, it's not good. Unfortunately, the kelp it just degrades too quickly. So we do use a synthetic line. And to date, if we can retrieve it, we will. But if adults grow from this twine, it's likely we wouldn't be able to retrieve it. But we haven't actually reached that stage yet. We've only used the hemp twine at our sites that degraded. So we'll see how it goes with the synthetic line.
Great. All right. Well, so anyone else have a question? You could unmute yourself or raise your hand. And Rietta, if you stop sharing, we can see the whole group. Anyone else have a question for Rietta? This is the first ever we haven't had a question.
You can always email me.
Nobody? Seriously? I mean, I think it's a little overwhelming, Rietta, because it's such it really makes me so grateful that we have GFA and the sanctuaries near us. You know, with Southern Mendocino Coast is included in the Gulf of the Farallones, so we get a little bit of Mendocino in there, but an organization of your capacity, it's so clear to me now after all these years, is what's needed to help, you know, really sort of drive these are such huge efforts and huge processes, and it really it's not something an organization our size can do just from the insurance and permitting alone, right? So having you guys take the leadership you have has been amazing, and the work I've been watching your work for a number of years, and it's you really are an environmental hero, if I have to say it's succinctly. It's not, this is
Well,
not.
thank you. I really appreciate that.
Yeah. You're getting a lot of thank yous in the chats. Oh, Wendy has a question.
Yeah, I have a question that Sheila mentioned earlier, and that's the counting the bull kelp on the beaches. That is part of our beach survey program, but we're not using you were doing way more than what we do. We're just counting whether they're adults, whether we see juveniles, and and whether we see reproductive spores or not. So do you think that's enough? Should we be doing more? What and also, like, is there a correlation between what's washing up and what's growing out there?
Yeah, that's a great question. And I would say we don't know yet. So these past few months that we've been doing these surveys is to lay a baseline. There is a lot of influence from waves and currents on where bull kelp or any rack might wash up. And so we don't really know the association yet between the restoration sites and the nearby beaches because there's so many different factors. But we're really kind of hoping that these data, when we start collecting the data from the hydrodynamic study, that it might reveal some interesting information, as well as when we're advancing our restoration work. So this is all pretty new for us, but we did want to be really robust in the information that we collect so that we could lay a pretty solid baseline and then continue those over time. So those are being managed by my colleague, Angela Zepp. So she has more information about them, but I don't know that you need to collect more data for the sites that you're doing. It's definitely always great to be able to compare across the region, but we're doing very fine scale because we want to capture that data adjacent to our restoration sites. And this is all new for us. So like we have a beach watch program, but it only counts like presence absence of beach rack. So I don't know. We'll be able to tell more in the future when we have more data. But it is a pretty big opportunity to be able to share information and data over time.
Yeah. Okay,
Thank you.
your questions have been pouring in since you've been talking. So Ken has asked if you've seen any sea otters in your study area.
No, we haven't seen any sea otters. And there's not very much sea otter presence on the Sonoma coast. The only time I've seen otters up in the northern area was actually in the bay, in San Francisco Bay. That was interesting. We have seen quite a few river otters, but I know obviously that's quite a difference. But I think we're kind of out of their current range in terms of their presence at our sites.
Okay, then Ken continues with, once you have realized success at restoration sites, so that's nice and hopeful there, how do you envision maintaining kelp growth in the long term from their restoration of albertine health and the potential return of the sunflower sea star? So let's start with, somebody also asked if you've seen any sunflower sea stars in your area, and then talk about the long-term kind of vision.
We have not seen any sunflower stars yet. I know there have been some sightings of sunflowers up in Mendocino, but in Sonoma, we haven't seen any. We've seen lots of ochre stars and giant spine stars, but no sunflower stars as of yet. Cross our fingers. And then in terms of maintaining growth over the long term, so this is where our collaboration with the federal entity as well as the state is really important. So the state of California right now is working on developing a kelp restoration and management plan. So they've developed these community working groups and a science advisory committee. And over the next couple of years, we'll be putting together this restoration plan. And that is going to set the framework for management response to kelp loss in the future. And I think that's where we need to focus a lot of those efforts, is because on the North Coast, we saw this, as I said, catastrophic decline of kelp and then no response for years. No one was doing anything about the kelp loss on the North Coast. And that's just crazy. It's insane because, there's such robust management of resources in California, natural resources, but kelp forest just had none whatsoever. The infrastructure didn't even exist. So the fact that all of this restoration work is happening now is amazing. It's exciting. It's wonderful that there's so much momentum, but it's late game, really late. So hopefully this restoration plan, as well as all of these other projects that are happening, they'll all be incorporated into a much more adaptive, responsive plan to be able to enable management to respond to kelp forest loss much more quickly. And then there's a lot of research to help us understand what causes kelp loss and what helps with kelp recovery, and then what's successful, what's not. So what we're doing is all new. Like, we're learning as we go. And you know, it is very exciting, but all of it is like, we kind of have to see, like, we have some of the experts in the fields advising the project and being a part of it. But even they don't know what's going to be fully successful or not. So it's a very exciting time, that will hopefully build a better framework in the future for responding to kelp loss events. Hopefully that answers your question.
Yeah.
I think we're
that's
going
fine.
to see kelp loss again in the future, but it's just a matter of how do we respond to it.
I think there is no great answer to that, but you gave as good an answer as anyone could give right now. I think we just got to keep trying. Okay, so somebody pointed out that Cal Poly was working on Humboldt Cal Poly was working on bull kelp spore culturing and outplanting. We got a science talk on that too. Have you been collaborating with their team at all up in Humboldt?
So we work, like I said, with Sonoma State University and Moss Landing Marine Lab. And they've been really leading the fields in terms of outplanting on the North Coast in our area in Sonoma and Mendocino. They also work up in Mendocino with the Nature Conservancy, and they've done outplanting at quite a few sites. We have not worked as much with Humboldt, but I'm aware of the work that's happened up there. And we're kind of all a part of the same community. So I think there's a lot of opportunities for us to share information, and what works and what doesn't. But we really kind of, to put it simply, we contract that out. So we pay Sonoma State University and Moss Landing Marine Labs to really be the experts on outplanting for our project. And they are. And they're amazing and fantastic. And they're helping us build that infrastructure so that we can lead outplanting in the future. And so that's our kind of main line of collaboration, is all of the really incredible work that they've done at their sites.
Okay, great. So Mary Glanville asks that since there's no bull kelp in the intertidal zones, are you looking at how urchin are impacting some of the other seaweeds in the intertidal? And if so, are you focusing on any particular ones?
Yeah, good question. So there's a range of intertidal algae that our Limpids program collects data on. As of right now, we don't have a specific species that we're tracking. What we're looking at is algal cover, so like percent cover, and that's all species of algae in the intertidal. But as of right now, that project is really focusing on urchin densities and then comparing that with the Limpids data. So it'll be really focused on percent cover. Hopefully that answers your question, but we don't have like one specific algal species that we're like, this one needs to recover more than the other ones.
Yeah, your surveys and work in the intertidal kind of piqued my interest. That's another something that we could potentially start to do more of here with our team of community scientists. I think that's another good opportunity to get more people involved. Especially when it comes to like, we're still struggling with how to collect for the aquaculture versus for restoration, because you have to collect so much more carefully. And if we started a program where we were, especially because so many of the abalone are in that shallow subtidal intertidal zone now, because that's where there is a little remnant seaweed left. It's an interesting idea to target that area as a little mini restoration project.
Yeah, I think it in the future will be another good opportunity to engage with volunteers to make a difference if people don't have like scuba certifications or are limited in some way in getting underwater. And then when we pitched it to the state of California, they were pretty excited about this type of approach because it's not being done anywhere else. There's no like intertidal restoration projects. So
Yeah.
this will be the first foray in that arena. So we'll see how it goes.
Yeah.
It's very exciting.
Rietta, we hope to have you back when you get done with another field season to see how you're progressing with all of these research initiatives. So thanks a lot, Rietta. Appreciate it so much.
Yeah, thank you so much for the invitation.
So again, thanks everybody for spending the evening with us, and Rietta, I'll see you in the next Zoom meeting.